All right. So, good morning, everybody, and welcome to this week's Cell Size and Growth Seminar Series. So, today we have two very exciting talks about the role cell volume and also cell volume asymmetry plays in development and differentiation. So, we're going to have Fiona Watt, who's going to talk to us about cell volume's role in regulating skin stem cell differentiation, and Clemens Cabernard, who's going to talk to us about cellular asymmetry in, I believe, the fly. So, without much ado, let's hand it over to Fiona.
Great. Well, thank you. Oh, someone else is sharing. You can request.
I have stopped sharing, so we should be good.
I'll try again. Uh, yes. Just go on full screen. Can you see that? All right.
Yep.
Continue
Yeah.
to
Looks
share.
great.
Yeah. Okay, great. I'm new to cell volume control, although actually the first paper I published when I was a postdoc pointed out that the cells we work on increase in size as they differentiate. So it feels very nice having the opportunity to talk about it again. So just to give you a brief introduction to the tissue we work with, it's the epidermis, it's the outer covering of the skin, and everything that I'm going to tell you about is related to human skin. And the reason I really like working in this tissue is its simplicity. It's made up of multiple layers of epithelial cells called keratinocytes. Um, the proliferation takes place in the basal layer of cells attached to an underlying basement membrane. This is the compartment where the stem cells reside. And when cells stop dividing, they become committed to differentiate. And then they go through a journey in time and space as they move through the upper layers, changing in gene expression and morphology to the outermost layer of the skin. This journey takes about 28 days in most body sites. And these outermost cells are dead cells, but they're exquisitely specialized to act as a protective covering of our body. And so, as these cells are shed, they need to be replaced by proliferation of stem cells in the basal layer. So, you have this beautiful balance between stem cell renewal and differentiation. So, how can we study human epidermis in culture? Well, it's because of a paper that was published more than 50 years ago by Jim Reinwald and Howard Green. And they showed how you can take a biopsy of human skin, disaggregate the cells into a single cell suspension, and plate them onto a feeder layer of 3T3 cells. And as you can see here, the stem cells start to attach and divide, and they form little colonies of cells. And over time, these expand, and you start to see large cells in the middle of the colonies. And these correspond to cells which are differentiating and have left the basal layer. So we're not dealing with monolayers here, we're dealing with multi-layered colonies. So we know that stem cells must persist in culture because this way of growing human epidermis has been applied in the clinic for many years as a way of making autologous sheets of epidermis to treat burn injuries. So, working on this system over many years and being passionately interested in stem cells, um, I and others have really thought a lot about what the aspects of a stem cell microenvironment or niche might be that influence a stem cell behavior. And we've summarized some of these interactions here. And then we've gone into a very reductionist system in a cell culture dish and tried to mimic these individual interactions. And what I'm going to tell you about today is what happens when single stem cells interact with an extracellular matrix that they can adhere to. So, over the years, a large number of experiments from multiple labs have shown that one strong stimulus for the cells to differentiate is if you prevent them from spreading. So, you can do that by disaggregating the cells and holding them in a viscous medium for 12 hours, then the suspended cells differentiate. You can capture cells on circular micropatterned islands On a 50 micron diameter island, the cells spread and remain undifferentiated, whereas if it's a 20 micron diameter island, the cells remain rounded and they do differentiate. And in experiments we did where we plated cells on polyacrylamide substrates of different stiffness, we found that on the soft substrates, the cells rounded up and they differentiated. So the observation that restricting cell spreading stimulates differentiation is very robust. Another robust observation is that in all of those contexts that I've described, if you inhibit Rho kinase, you can block differentiation. Of course, Rho kinase has many functions as shown in this schematic here, but it is in particular associated with cell adhesion, bifocal adhesions, and by the interactions with the actin cytoskeleton. So some years back, we wondered in the lab if there was a way in which we could stimulate differentiation that did not involve the cells rounding up. And we did a collaborative project with Jan de Boer's lab in the Netherlands, where we screened more than 2,000 different pattern substrates, looking for substrates that affected differentiation of spread cells. And these scanning EM pictures here show two substrates that we have worked with subsequently. This substrate, which we call S1, consists of irregular circular pegs, and this one called S2 consists of regularly patterned triangles. And if you look down here, this is qPCR where we're measuring expression of differentiation markers. You can see on the S1 substrate, differentiation is stimulated, and on the S2 substrate, differentiation is actually inhibited relative to the flat control. We have some scanning EMs which show very nicely, this is the work of Misana, who's a postdoc in my lab, show very nicely how the cells interact with these substrates. So here's the S1 pillar substrate. You can see two separate cells. And if you look at the edge, you can see how the cell appears to be exerting force on the pegs. And I like this image here because you can see the pegs don't penetrate the cytoplasm, but the cytoplasm over the pegs is very thin. So cells differentiate on this substrate. And as you can imagine, we were really disappointed to find that even though the cells are differentiating while they spread, we can still block differentiation if we inhibit Rho kinase. And this is a flat substrate, the S1 substrate, the S2 substrate, and you can see the inhibitory effect on differentiation. And that turned us to our attention to this second substrate, S2, which, to be honest, we weren't really interested in at first because we were wanting to look at differentiation. But you can see here where we measure the percentage of differentiated cells that differentiation is actually inhibited on S2. And when Misana looked at the interaction of these cells with the substrates, she got the images that you can see here. So in some cases, the spread cell's nucleus is actually quite distorted and embedded down between the triangles. And there are other examples where you can see the nucleus above the triangle. So how is it that this substrate is actually inhibiting differentiation? My then PhD student, Sebastian Zeil, who is now a postdoc in Cambridge, noticed that on the S2 substrates, the volume of the cells was lower than on the flat or the S1 substrates. He made these measurements using confocal imaging, as you can see over here. And here we see the reduction in both cytoplasmic volume and total cell volume. So this was of interest because, as this slide from Misani shows, we knew that as cells differentiate, they get larger. So it got Sebastian wondering whether cell volume might be an important regulator of differentiation. So just to show you some examples here, if you measure the ability of cells to form colonies, there's this curve that you can see, whereas with increasing cell diameter, you see decreasing colony forming efficiency. So what Sebastian did was to look at this paper from Dave Weitz's group at Harvard, where he had used polyethylene glycol and distilled water to change the volume of mesenchymal stem cells. And he tried these treatments out on oops, on our cells. And he found that sure enough, if he added polyethylene glycol to the medium, he could reduce the volume of the cells. And if he supplemented the medium with distilled water, he could increase cell volume. So then, of course, the question was, does this have any effect on differentiation? And the results were really spectacular. In every assay that we did, changing cell volume affected differentiation. So if you look at distilled water, you can see on flat substrates, the percentage of differentiated cells increases. On micropatterned islands, where normally on a large island the cells are spread, if we treat with distilled water, they differentiate. If we Conversely, if we add polyethylene glycol to cells in suspension and probe for the differentiation marker involucrin, you can see differentiation is inhibited. And on those substrates that I described, we can stimulate differentiation with water and we can inhibit it with polyethylene glycol. And on the S2 substrates, distilled water stimulates differentiation. These effects that we're seeing are reversible, and that's shown in two experiments. So in this experiment here, Sebastian treated the cells with polyethylene glycol to inhibit differentiation and then washed it out. And you can see that actually differentiation is restored, and the proportion of differentiated cells is actually higher than the controls. And in this experiment, which Misana has done, she wanted to know how long she could treat the cells with distilled water and they would still without them differentiating. So she treated for the number of hours that you can see here and then allowed the cells to recover. And it looks as though the point of no return in terms of being exposed to distilled water is about 8 hours. So this gives us interesting clues about underlying mechanisms. So on the subject of underlying mechanisms, this is very much where we are at the moment. One of the things that we did was to do some bulk RNA sequencing, looking at cells plated on the S1 and S2 substrates, comparing the 1 hour and 4 hour time points when the cells don't differentiate, and 1 hour and 12 hours where they differentiate on S1 but not on S2. And so you can see there are a number of candidate regulators like aquaporins, intracellular calcium ions, and ion channels. And in really initial experiments, we tested the effects of two inhibitors. One was aquaporin 3 inhibitor, and you can see very nicely here that it blocks differentiation induced by distilled water. And in the same way, if we treat with this intracellular calcium ion chelator, BAPTA AM, we can block differentiation. Conversely, if we treat with gadolinium ions, we didn't see an effect. So to summarize what I've said, in these experiments that I've shown you, we can see that treating with polyethylene glycol reduces volume and inhibits differentiation, whereas if we add distilled water, we increase cell volume and we stimulate differentiation. We're working actively to try and identify the underlying mechanisms. And a question that I've personally found really interesting is whether or not there is synergy between cell spreading and cell volume. And the reason I think that is interesting is if you think back to the histology of the epidermis, you have the stem cells attached to the basement membrane. And you can imagine that as they get bigger because they've been out of cycle, the area that is able to accommodate that increased cell volume is reduced. And that could trigger the onset of differentiation. That's speculation, something that we're really interested in. But all in all, it's been a very interesting journey for us to start to think about cell volume and differentiation. Last slide is the most important. It's a big thank you. You can see Mesana here, who's done all of the cell volume work in the lab at the moment, other members of the lab, Sebastian, and our funders. So with that, I'll say thank you and stop sharing.
Thank you so much, Fiona. Yeah, it's such a sorry. Just for everybody in the chat, if you have questions, please put them in the chat in writing, and I will basically go through them one by one to um, yeah, and present them to Fiona. If I could start with a question, which is, have you ever essentially, I wonder, I guess to get at the question of whether cell volume is kind of this controlling variable or the most sort of close correlate to the differentiation propensity. Have you essentially plotted all of the experiments, from all these like aggregate all experiments and just plotted cell volume on one axis and then percent differentiating on the other? And barring like batch effects and so on, but to see if essentially the in between cell spreading and all these like ionic and also crowding experiments, whether essentially they are controlling volume, which is kind of this integrating variable, and then that is essentially upstream of something that's being reading out cell volume and then subsequently.
Yes, I mean, years ago, I did some experiments where I treated the cells with a drug that stopped them dividing and left
Yeah,
them for weeks and asked,
yeah,
and they got bigger and bigger. That was
yeah.
not a sufficient stimulus to differentiate.
I
So
see.
increasing size is not enough. The question about plotting cell volume versus differentiation propensity is one that actually Misana and I were discussing this afternoon. Um,
Hmm.
I think I showed you some the old experiments from the 1970s and 80s correlating cell volume with colony forming efficiency.
Hmm.
We probably did some measuring a differentiation marker. My feeling is that it is a continuum. I don't think it's a kind of a switch where you reach a certain size and differentiate. And we know
Hmm.
that when you put cells in culture, pretty much any cell, they immediately get bigger and they start making more protein. So I'm not
Hmm.
saying the cells aren't measuring their cell volume, but I do know that simply stopping them dividing, letting them get bigger is not a sufficient stimulus.
No, I see.
And
Okay.
yeah, yeah.
Okay, thank you. Okay, let's there are lots of questions in the chat. Let's start with Kiki.
Shall I read it out? Oh,
I think he will read it out usually,
Kiki, you
unless he
read it
doesn't
out.
want to.
It's your question.
I could read it out
Does
if
there exist a gradient
yeah.
from the inside to the outside of the skin, such that the outside somehow resembles distilled water more than the inside? You had presented the result that distilled water promotes differentiation, presumably by letting more water into the cell and changing the osmotic situation in the cell. But that sounds counterintuitive in context, because I would imagine that the outside of the body is drier than the inside of the body. And so I would expect that the closer you get to the outside environment, the less the osmotic environment would resemble distilled water.
So I think it's a an interesting question. It's reported that there are gradients within the epidermis, for example, in the intracellular calcium ion concentration. But as to whether the outside of the skin resembles distilled water, you have to remember that those outer layers of the skin are heavily cross-linked insoluble proteins and lipids, and they're designed that when you go out in the rain, you don't swell up because water doesn't penetrate that surface. So yes, there are gradients. I'm not sure if people have looked at water. There are also gradients in expression of some of those genes that I mentioned, which regulate cell volume control. But the surface of the skin is a really important barrier for microorganisms as well as water. So I don't think it's as simple as that.
All right, thank you, Kiki. Next we have a question from Bruce.
Hi. When you change cell volume with distilled water or PEG, over the short term, you kind of expect proteins to be concentrated or diluted, and so intracellular protein concentration to change. Over the time courses you were discussing, the cell would have a chance to push those back to some homeostatic level. So you could imagine the protein concentrations have or haven't changed in your experiments. Do you have any insight as to whether they've changed or whether protein concentration might be the thing that matters?
We haven't looked at that. It's a good point. Three things that might be relevant. One is that Misan has done some initial labeling with reporters of protein of molecular crowding, and it does look as though there are responses to PEG or distilled water. So I wouldn't be surprised if something like that is happening. But
So in the intuitive direction, so
um,
less crowding in water and more crowding in PEG.
I think that that would be right, but I don't want to be quoted on that because I would need Miss Sana to say for sure. But what we do know is that the volume of the cells, if you treat with distilled water, they're able to return their volume back to normal over time. They have good volume control. And then the other thing about protein concentration, which is a special thing about the epidermis, is that as you differentiate, you make more and more elaborate keratin cytoskeleton. And so the total protein content of the cells will go up as they differentiate. But I think it's really interesting to look at these issues and look at subcellular distribution.
Okay, thanks very much.
Thank you.
All right, thank you, Bruce. Next we have another question from Kiki. If you want to ask about the outer layers of the skin.
Thank you for your patience. This is less relevant to the immediate subject of the presentation, but I have been wondering about it ever since the diagram at the beginning. When skin cells die at the outer surface of the skin, do they undergo apoptosis or anything like that? Is there instead some specialized process to transition from a living cell to a non-living interlinked element of a protective layer?
That's an excellent question. So there are elements of the differentiation process which cells have in common with apoptosis. So, for example, activation of caspases, changes in the plasma membrane. However, actually, when you look at markers of apoptosis, it is rare in the epidermis. The only regulated apoptotic events that I'm aware of happen during the hair growth cycle when the hairs are stop growing and are shed. So the process of differentiation has elements and like activation of transglutaminases, which are reminiscent of apoptosis, but the time course is different. And something I've always wanted to look at is that the way the nucleus is lost in the outer stages of differentiation is quite different from what you see in an apoptotic nucleus.
All
So
right, thank
we can
you.
perhaps think of it in a weird way as differentiation into a cell fate that happens to be dead.
Yes, it's like life, isn't it?
Mhm.
Since you mentioned it, why is the nucleus lost at all?
Well, in some skin diseases like psoriasis, it's not lost. It's called hyperkeratotic cells. And in that case, I think the cells are moving so fast that there isn't time to lose the nucleus. Um, why is it lost? So it doesn't have to be lost, but I think it's just incompatible with being in a cell which no longer has any mitochondria or other cytoplasmic organelles. And the whole thing just becomes this um, beautiful network of insoluble proteins and lipids.
All right, thank you, Kiki. Next we have a question from Simone, but I believe it was already answered about molecular density changes, which basically was part of Bruce's question. So, sorry, Simone. Let's skip to Matthew's question.
Thanks. Sorry if you said this and I missed it, but kind of what specifically is it about the S2 substrate that's different to the S1 substrate that you think results in the reduced volume? Is that an obvious consequence of the way it makes the cells spread, or that's unclear?
I've got no idea.
Okay,
I
fair
mean,
enough.
it's looking at cells on the S2 substrates is a horrible thing because you can imagine when the nucleus has gone down between the triangles, cell does not look great. But we were because the cells get bigger as they differentiate, in those measurements, we were very careful to look at a 4-hour time point before there's any differentiation. So, and we're plating cells which are the same size. So I suppose the question would be, is there any increase in cell volume at 4 hours in the control substrate and not on S2? I don't know.
And,
I mean, to be honest, we made that substrate because we thought we'd identified another pattern that stimulated differentiation, but the scale up that we got wasn't the same shape, and we just noticed that the cells were suppressed.
and then something else that I was wondering is like the extent to which these substrates or different conditions are affecting differentiation and stem cell renewal? Are the cells just doing nothing? They're not choosing to differentiate, and they're also not proliferating, or are they proliferating and not differentiating? Does that make sense?
Yes, epidermal cells are unusual in that they can differentiate at any phase of the cell cycle. And so you can actually, if you look in a lesion of psoriasis where there's a lot of proliferation, you can get cells which are in cycle but have switched on differentiation. Normally, what is happening is within the basal layer, you've got stem cells which don't actually divide all that often, but the cells which are going to leave the basal layer have been out of cycle for a while. We call them then committed to differentiate. And as they execute the differentiation program, they simultaneously downregulate their integrins, upregulate cadherins and desmosome proteins. And so they're more attracted to sticking to the suprabasal cells than the basal cells. But that commitment process depends on a network of interacting protein phosphatases that negatively regulate the ERK-MAP kinase pathway.
Okay, thank you.
All right, thank you. So in the interest of time, I'm gonna move on to Jin Rong's question.
Yeah, thanks. Here's it feels like the calcium response is quite crucial in the process of start activating the differentiation process. So I'm curious if uh, um, uh, the calcium related programs are actually at the downstream connected to some of the transcription factors are off. Um, when it's in the stem cell state, well, it's gradually turned on when the calcium response is on, when you have the change in the cell volume. Yeah, the sets of the transcription factors that control the differentiation. Yeah.
Yeah, the if you reduce the extracellular calcium ion concentration, you can prevent the cells from forming cell-cell contacts and making a multilayer, but the cells can still differentiate. So they can differentiate regardless of the extracellular calcium concentration. As for intracellular calcium ions, there are suggestions of a gradient. We haven't looked specifically at calcium responsive transcription factors, but I would say that most of the differentiation program depends on upregulation of AP1 transcription factors. I'm not sure if they're calcium responsive.
Okay.
Yeah.
Yeah, thank you.
All right, thank you. Let's take one last question from Jish.
Hi, yes. If I recall correctly, knocking out the aquaporin prevented differentiation in your experiments. And I want to know, does the aquaporin knockout prevent the cells from increasing their size in DI water, or does it prevent differentiation despite an increase in cell volume?
That's a great question. It wasn't a knockout, it was a drug that we used. And to be honest, we just didn't look at whether it affected cell spelling, but we could and should look at that because it's part of a series of experiments that Misana is doing now to try to track the pathways and the genes that are important there. And it's interesting. It's a whole other world. I had no idea there were so many aquaporin genes.
Thank you.
All right. Thank you again, Fiona.
My pleasure.
Thank you. Okay, next we have Clemens from University of Washington.
All right. Okay, so let me see. All right, can you all see this?
Yep, looks great. Thank you.
Okay. Well, thanks a lot, Mimi and Matthew, for this kind of invitation. This is a great opportunity to tell you guys a little bit about what my lab is doing. And usually we don't really think so much about cell size and growth. So this is a great platform to also bounce off some ideas and see what you guys think about this project. So my lab works on asymmetric cell division, which you probably all know is a very important process to form differentiated sibling cells. So it happens in a lot of developmental contexts during embryogenesis, leaf development, skin differentiation, and whatnot. And obviously, stem cells are one cell type that utilize this division mode to not only self-renew or recreate or reform the stem cell, but also at the same time forming different sibling cells, which are important for different functions in the body. Now, we are using Drosophila neural stem cells or neuroblasts, also called neuroblasts, as a system because we can use live cell imaging to really look at the dynamics of these cells. As we can see in this movie here, all these large cells, which are rapidly dividing, are neural stem cells. And what they do is they form these smaller cells on one side, which will then go on to differentiate into neurons or glia. And so we study this process in the developing larval brain, mostly using fly larvae in order to not only do live cell imaging, but also study the dynamics of this process. So it's been known that these neural stem cells are intrinsically polarized. A lot of labs before us have shown this. We can visualize this polarity axis using apical PAR complex proteins shown here in green, which nicely localize to one side of this neuroblast in mitosis. And as this neural stem cell is dividing, they stay or remain in the cell-free neural stem cell. On the other side of this neural stem cell, we see the cell fate determinants shown in red, which nicely segregate into the smaller sibling cell, which will then go on to form a ganglion mother cell that divides one more time to form either two neurons, a neuron or glia, or two glial cells. So this is just to show that at pretty much all times, we precisely know where the apical and or basal cell cortex is located in these cells. Now, another interesting feature of these neural stem cells is they divide asymmetrically by size. As this movie shows, we get this large cell-free neural stem cell, while at the same time, a much smaller sibling cell on the basal side of this cell. And so when we started working on this process, we focused a lot on the dynamics of the actomyosin cytoskeleton. Here we're looking at myosin or non-muscle myosin in green, which very dynamically relocalizes during cell division from this uniform cortical localization towards the cleavage furrow. And so I'm not going to show you all the data, but what we actually learned over the years is summarized in this cartoon here. These myosin dynamics are essentially regulated by two spatial temporally separated molecular pathways. On one hand, polarity cues starting with pins, also corresponding to LGN or AGS3 in vertebrates, is recruiting a kinase called protein kinase N, which most likely downregulates kinase, Rho kinase, which we have heard before from Fiona, which then leads to a loss of activation of myosin in this apical cortex, and that results in a basally directed cortical flow whereby myosin flows towards the cleavage furrow. So this happens first in early mitosis, and shortly after, about a minute later, the pathway continues through a spindle-dependent pathway, which keeps activating Rho at this basally shifted position where the cleavage furrow will ultimately form through microtubules, activating Rho, which then pulls myosin towards the cleavage furrow from the basal side. So we have a second flow phenomena from the basal to the apical side, and the interplay and the temporal separation of these pathways ensures that the cleavage furrow is essentially shifted towards the basal side, which then allows to form a larger apical and a smaller basal cell. Now, while this is all interesting, one question that has always been open is, why do these neural stem cells do this? Why is it important to invest so much energy and regulation in the formation of two unequal sized sibling cells? And so when we look into the literature, there is a couple of examples that can maybe shed some insight into why this could be important. So in worms, for example, a similar cell type has been shown to divide asymmetrically by size, whereby the largest cell will always form a neuron versus the small cell is undergoing apoptosis. And if through certain manipulation, the cell type is being forced to divide symmetrically, two neurons will form. In humans, cell culture, mostly cells divide symmetrically by size, but if these cells are being forced to divide asymmetrically by size, then the smaller cell is undergoing apoptosis versus the larger cell keeps dividing. And so in our system, we thought where we have this very clear binary cell fate readout where the large cell is self-renewing itself to remain stem cell versus the small cell differentiates, we thought maybe by manipulating the cell size ratio, we could learn something about how cell size is connected to cell fate and differentiation. Now, how do we do this? So given this molecular insight we have about the regulation of this process, we developed this tool called a nanobody, which is shown here in brown, which is essentially a single chain antibody with a high affinity for GFP. And so what we did is we, on one hand, functionalized this nanobody with a kinase domain from Rho kinase. And on the other hand, we tethered the nanobody specifically to the apical side of these neuroblasts using an apical localization domain from inscrutable. And so the idea being, if the only protein carrying a GFP tag is myosin in these cells, then the nanobody should essentially bind to the GFP tag and maintain an activated version of myosin on the apical cortex because it continuously phosphorylates myosin that's being trapped on the apical side. And so essentially what we were hoping this would do is would essentially allow us to change the flow pattern of non-muscle myosin and therefore physically invert these divisions so that we get a small apical and a larger basal cell. And so this has been work from a former postdoc, Chantal Robinet, which actually worked really, really well. So she showed here in this movie where we have apical on the top, where we see an enrichment in myosin, that if we maintain activated myosin on the apical side, this indeed is sufficient, not always, but in some examples, to invert these physical asymmetric divisions. Now, obviously, the question is, what are the respective fate outcomes of these cells? What is now the small versus the large cell going to do over time? And so this, before I say this, one thing we also want to make sure is that intrinsic polarity is actually not changed. So in normal fly neural stem cells, as I've shown you before, Miranda basal cell fate determinant nicely segregates into the smallest cell. And so even if we happen to invert these divisions, as shown in this example, where neuroblast is undergoing an inverted division, Miranda or basal cell fate determinant still segregate nicely into this, in this case, the larger basal cell. So intrinsic polarity is not changed. although we change sibling cell size to symmetry, so we uncouple polarity from cell size. Now, a grad student in my lab, Melissa Delgado, was really interested in asking, how does this affect fate and cell behavior? And so to answer that question, she essentially did long live cell imaging to really see whether we see any changes or differences in cell proliferation behavior. And so this is a standard wild-type neural stem cell going through a first asymmetric division here at 004. And then 3 hours, 41 minutes later, it's going through a second asymmetric cell division, and it would continue doing so if we would just record this neural stem cell a lot longer. So while 3 hours is quite long for a normal interface, neural stem cell interface, we can find cells that sometimes do this. Now, this is an example of an inverted division. First, it forms a large basal cell and a small apical cell. And in this case, the larger basal cell will divide again after 3 hours, 20 minutes, which is hard to see here, but it divides in an asymmetric fashion again. So in this case, the lineage is actually flipped. We get a smaller cell on the top, a larger cell on the bottom, and the large cell will undergo another division with an interface that's comparable to wild type. Interestingly, we actually also find symmetric divisions when we express an antibody. So here, for example, is a neural stem cell. Mice is being trapped on the apical side. A first symmetric division happens at this point. 2 hours later, the apical cell undergoes a division, which appears to be symmetric again. And then the apical cell, one more time, undergoes a symmetric division before the larger basal cell that was created in the first division here is undergoing division. So this lineage tree looks like this. We have a succession of symmetric divisions whereby the apical cell divides 2 more times, and after only 13 hours, the basal cell is undergoing symmetric division. So when we analyze all these lineages, we can essentially ask whether there is a pattern. And the first question we were asking is, in cases where we have an inverted asymmetric division with a small apical and larger basal cell, we can ask which cell is more likely to divide, or in other words, when we looked at the movies, which cell did we find dividing more often? And so the results are pretty clear. In almost all cases, we see that the basal cell is dividing again, but only in 2 instances, so less than 40%, we find that apical cells were able to divide within the time of recording. Equally, of these 2 cases, if you're asking if we have both cells dividing, which one of these 2 is dividing first? And it's in all cases, although the numbers are really small, we find that the larger basal cell has a much shorter cell cycle compared to its apical sibling. Now, if we consider symmetric cases where we have 2 equal-sized cells and ask which one is more likely to divide, we actually find that the apical cell in all cases divided versus only in about 30% of the cases, the larger basal cell was dividing within the window of recording. And in this case, if we ask which one is faster, it's always the apical that divided before its basal sibling in cases where both siblings were dividing. So there seems to be not only a cell size relationship between proliferation and cell size, but also a position-dependent effect, as shown with these symmetric divisions. Now, we wanted to know, if we're looking at actual cell fate markers, what is the outcome of these divisions? And so Ian Hurstler, another postdoc in my lab, engineered this deadpan tomato transgenic line at the endogenous locus. Deadpan is a neural stem cell marker, which is often used to assess what the fate of these cells is. And so in this case, this is a wild type division as a control. We start with a nice large deadpan positive cell. This neural stem cell is undergoing an asymmetric division, recreating the stem cell while forming a smaller sibling. And while deadpan is also segregating into the smaller cell, it will fade over time, leaving behind one large deadpan positive and a smaller weakly deadpan positive GMC. Now we could ask, what about symmetric divisions? How does deadpan segregate and ultimately is retained in these cells. Here we have a symmetric division after nanobody expression to equal-sized nuclei, deadpan positive. Deadpan fades a little bit, which is to some extent also due to bleaching, but both siblings essentially retain equal amounts of deadpan after these symmetric divisions. We also have inverted cases, as shown in this specific example. This neural stem cell after nanobody division divides inverted, whereby we get, again, a small apical cell, which happens to be deadpan positive, and also a larger basal cell, which is also deadpan positive, which was kind of surprising because we expected that maybe in this case, we only have one cell that expresses the neural stem cell marker. So to summarize this first part of what I just told you is wild-type neural stem cells repeatedly divide asymmetric, whereby the top cell is self-renewing itself to maintain stem cell fate, versus the smallest sibling is essentially undergoing differentiation and has a much larger cell cycle compared to the larger cell. If we induce symmetric divisions whereby polarity is still normal, we can observe that both cells, in most cases, actually in all cases, are positive for the stem cell marker, but the apical cell clearly has a much shorter cell cycle and is more likely to divide compared to its basal sibling. And when we look at the inverted divisions, in some cases we get small apical cells which are deadpan negative, but we also get small apical cells which are deadpan positive, which if they divide, which is not very likely, they have a much larger, longer cell cycle compared to their larger basal siblings, which are also deadpan positive. Okay, so now we wondered, how does that actually affect neurogenesis? And so these wild-type neural stem cells essentially create all the differentiating siblings that occur in these larval brains. And so the only division mode they undergo is asymmetric, which then forms different types of differentiated siblings in these lineages. And so here, this is a cartoon of a fly neural stem cell lineage. Stem cell is usually arranged at the top with sibling cells that differentiate, clustering on one side of these neural stem cells. Now, we also introduced or used a mutation which I haven't introduced before, which is called mushroom body defect, which to some extent, about 15% of the cases, we can also get symmetric divisions, which I have shown in earlier work gives rise to 2 equal-sized neural stem cells. So the question really is, in this case, although we get an increase in the stem cell pool, how does that affect differentiation? And now the third case in these nanobody brains, in addition to normal asymmetric divisions, we have symmetric divisions creating 2 equal-sized stem cells, as well as these inverted divisions, which have smaller and larger deadpan positive cells. And we could ask the same question, whether this increase in the stem cell pool has an effect on the number and type of differentiated siblings. And so what we used here is a brand new technology I think nobody has heard before, which is expression profiling using RNA-seq, whereby we looked at wild-type brains, compare them to mushroom body defect, where we have a certain percentage of symmetric divisions, and nanobody brains, which also have inverted asymmetric divisions, essentially asking, do we see a change in the number and types of cells? And so as you look at these UMAP plots, you hopefully see that there's not a whole lot of differences between these different conditions, which sort of surprised us because we were expecting maybe certain cell types will be missing, or maybe we see the creation of a new cell type that doesn't actually exist in wild type, but that was not the case. However, when we looked at these clusters in more detail, I'm not going to tell you what each number means, but when we looked at the stem cell pool here, which is represented by clusters 15 and 19, we actually see that there is a change in the number of stem cells in mother nanobody compared to wild type. Consistent with the movies I showed you, we actually see 1.5 to 2-fold increase in the number of stem cells in mother nanobody compared to wild type. Now we can use this RNA-seq data sets to essentially ask, how does that affect differentiation? Having more stem cells could mean we should also get more differentiated siblings. And so in wild type, one marker we can use to essentially assess all types of neurons is prospero. So all the sibling cells that are being produced by the parental neuroblast or neural stem cells should be prospero positive. And so comparing RNA-seq data with actually immunohistochemistry, we lineage by lineage counted how many prospero positive cells we find that are being produced by a parental neuroblast, and the numbers correspond pretty or are very closely between RNA-seq and immunohistochemistry. And essentially what this graph shows you is that in either case, we see a reduction in the overall production of prospero siblings compared to wild type. So despite the fact that we have more neural stem cells, overall we form fewer prospero positive siblings. Now, when we look into ganglion mother cells, so the youngest or the first cell type that's being produced by these neural stem cells using a marker called grainy head, we see a similar trend whereby both RNA-seq and immunohistochemistry suggest that there is a reduction in the number of grainy head positive ganglion mother cells being produced, despite the fact that we have more neural stem cells. And the same applies to newborn neurons labeled with Hay, where we also have a reduction in these cell types compared to wild type. Now, this suggested that maybe this can all be explained by simply a reduction in proliferation rates. So we looked into phosphohistone 2, a marker for mitosis, and we find dramatic about a half reduction in proliferation rate by half in nanobody brains compared to wild type, whereas there's also significant reduction in mud. So what essentially this shows is while we have a reduction in proliferation rates in nanobody in mud, this impacts how many differentiating siblings this can form. However, we still wanted to know whether that actually makes sense in terms of absolute numbers. And so we teamed up with my colleague Neda Bagari here in the department, which is essentially involved in mathematical modeling. And we built this base model, which essentially recapitulates all the basic features of neural stem cells. In wild type, the model involves regular asymmetric divisions where these neural stem cells form GMCs, which divide symmetrically to form immature neurons, which then mature into mature neurons and at some point leave the system. And then we build in symmetric divisions, which essentially double or should double all these different cell types. And so using all the measurements we have obtained from our wild type live cell imaging data, when we build a base model, we actually see that there should be a dramatic accumulation of both neural stem cells, GMCs, immature and mature neurons. So there's some mismatch between the model and what we actually see in vivo, suggesting that there's some type of regulation we haven't considered yet. So one thing we know is that these neural stem cells, when they divide asymmetrically, they usually have to grow back to their threshold or original volume before they can divide again. However, in symmetric divisions, because they divide symmetrically, they usually end up smaller compared to their parental neuroblasts. And so it would essentially take them longer until they achieve this threshold volume. So we modeled whether starting with a smaller population of neural stem cells, whether this could essentially prolong cell cycle and therefore lineage expansion. And indeed, by simply modeling this regrowth rate, we could see that in mud and nanobody, we can recapitulate the numbers in the model we see in vivo. We can also change this model in the sense that they don't have to grow back to their original threshold or parental volume, but could divide earlier. Even in this case, because we start with much smaller cell after symmetric divisions, it will bring back down the numbers compared to wild type. Now, a second type of regulation, which is maybe less intuitive, is sort of a self-inhibition or neuroblast repression model. So assuming that the minute we get a symmetric division and the system realizes there are now more than one neural stem cell, maybe they could either repress each other or one cell could repress itself in its ability to divide again. And using this type of model, we can also recapitulate the numbers in cell types we see in vivo. Now, obviously, the question is, what are the mechanisms for these two volume-based or self-repression mechanisms? And so while we don't quite know how volume and cell proliferation and lineage expansion are related, we maybe have some insight into the self-repression mechanisms. And so to this end, we imaged Prospero, which is a self-differentiation marker, as I've shown you before, which actually segregates asymmetrically, as this movie should show you. So here's a wild-type neural stem cell label in gray, Prospero in green. In mitosis, Prospero usually is forming a basal crescent, which then segregates into the smaller GMC, enters the nucleus, and so we end up with a smaller cell that is then starting a differentiation program. So this is the normal wild-type progression of Prospero. And so we assumed that maybe if we have symmetric divisions, Prospero could somehow change the fate and the cell behavior of the two siblings. And so here, this is symmetric division in mud. We're starting with one parental neuroblast, divide symmetrically, giving rise to two neural stem cells. And as you can see, when we look at nuclear Prospero levels, we now get an asymmetry where one neural stem cell has more Prospero than its sibling, as shown here. So indicating that despite they divide symmetrically by size, they're still slightly asymmetric in their molecular composition right after division. And we also find cases like this one here. This is another example of a symmetric division where a neuroblast divides symmetrically by size, as shown here, so ending up with two equal-sized sibling cells. Prospero looks like it's segregating symmetrically, and then in this case, we have weak Prospero, very low levels in both these nuclei, indicating that they either don't have enough Prospero compared to wild type, or they still have levels that differ from the actual wild type case. So to summarize all this and put this sort of in a model, the way we think about this right now is, this is shown here. These wild-type neural stem cells divide asymmetrically by size, whereby the large cell remains more proliferative, repeatedly forming smaller differentiating ganglion mother cells, which then go on to form all the different cell types in these lineages. Now, if we force these neural stems to divide symmetrically or asymmetrically along this normal polarity axis, we still see that one sibling is inheriting all the cell fate determinants, and these lineages are correspondingly much, much smaller because they either don't have the necessary volume or inherited some of these cell differentiation markers that drives them into differentiation. And similarly, if we divide symmetrically, perpendicular to the polarity axis, we also induce asymmetric segregation of basal cell fate determinants, which then affects lineage progression and the number of differentiating siblings in these lineages. So it's both a combination of volume and cell position that determines to what extent differentiating siblings are being produced, despite the fact that we have supernumerary neural stem cells in these cases. Now, on my last slide, I briefly want to mention the broader impact of this. So obviously, there are certain neurodevelopmental disorders, such as microcephaly, which give rise to this small brain phenotype. And asymmetric cell division has been involved in this process numerous times, because most of the loci that are involved in microcephaly actually encode for genes, many of which are involved in spindle orientation. And so the model has been that early on, these neural stem cells first have to amplify the stem cell pool before they switch towards asymmetric cell division to form all the necessary neurons in these developing brains. However, if that switch happens to be prematurely because of spindle orientation defects and therefore a premature switch towards asymmetric cell division, this could deplete the neural stem cell pool, or the stem cell pool could not reach a sufficient size, which impacts the number of neurons. However, our work suggests that despite having a large enough or even an increased neural stem cell pool, we still don't form sufficient numbers of differentiating siblings, which could raise some questions whether microcephaly could also have other causes. All right, so at the end, I would like to thank my lab. Most of the data I've shown you was obtained by Melissa Legato, former grad student, Ian Hertzler, a current postdoc, as well as Ilara and other former lab members. And of course, my collaborators, Sophia and Neda, were instrumental in this work as well. Thank you very much.
All right. Thank you so much, Clemens. Yeah. So folks in the room, please leave your questions in the chat. I would like to start with one, which is that you have this, from the mathematical modeling, it was clear that it was sufficient to, if you could control growth rate in a size-dependent manner, that you would get essentially the suppression of the exponential growth, right? Do you see that in your data? You have all these nice like live cell imaging. I wonder if this is something you can kind of systematically mine. I don't know how many neural blasts and neural stem cells you can actually image this way.
Yeah. No, that's a great question. So yes and no. I mean, so what is obvious from, let me answer this differently. The model really allows us to model these cell divisions over a much longer time span, which with live cell imaging is really, really hard to do. The longest we can really image for these cells is maybe 15 or 16 hours. And even then, we sometimes, because of photo damage and nutrition depletion, we see a slowing of the cell cycle in photo damage. Now, one thing even if we image on a shorter time scale or shift the developmental time window, we actually see in nanobody a very clear reduction in these proliferation rates. We can't really measure growth rates very accurately because we can't really measure volume very precisely. So measuring growth rates is difficult. Proliferation length or cell cycle length is easier to do. And even in mud, where we have symmetric divisions, and although they're still fairly proliferative, overall we see a reduction in their cell cycle length compared to wild type. So what we think what happens is this accumulates over time. The minute we get even more neural stem cells, the cell cycle length is progressively getting longer.
I see. Yeah. So I guess so do you have to dissect out these?
Yeah.
I see.
Yeah. And then one other thing I should say is we think there is a threshold. So even if you look in wild type range, there's quite some variation between neuroblast size or neural stem cell size and their corresponding cell cycle length. And actually, there is no real correlation in wild type between neural stem cell size and cell cycle length. We can have larger neuroblasts, which a long cell cycle, and smaller neuroblasts with a shorter cell cycle. There's no real correlation. However, the nanobody data clearly shows us there must be a threshold, because the minute they fall below a certain size, their cell cycle dramatically increases. Where that threshold really is, we don't know.
I see. Yeah. And I think that was so that kind of actually goes into Matthew's question.
Yeah,
which
you more or less covered exactly. As you were speaking, I had the same thought that you then answered as I was typing my question. So thank you. That's great.
Yeah, which is what so people who aren't reading, Matthew was asking if there's cell size homeostasis in the data. So essentially coordination between cell size and cell cycle duration.
Right.
Okay. Next we have a question or several questions from Kiki.
This time I place all my questions together to reduce the potential for disruption.
Thank you.
1 of 3. After a symmetric division which produces 2 stem cells, do the stem cells stick together, or do they separate to each form their own structure consisting of a single stem cell attached to its differentiated siblings?
Yes, that's a very great question. We actually were discussing this just 2 weeks ago, because obviously the model implied the self-repression mechanisms somehow could imply that they need to stick together, right? And actually, me, what they do, so we don't really see them separating from each other. They somehow maintain a shared surface as soon as they divide symmetrically, even if they do this multiple times. So they're usually clumped together in a little mini clones and then continue dividing with reduced rates from there onwards.
If that cluster of cells has a structure that assumes that it contains exactly one stem cell, does this break anything now that there are suddenly two?
What do you mean by break anything?
That is, you showed several cartoon images toward the end in which you showed these structures that supposedly result from symmetric division,
Right.
where there are now two stem cells in place of one.
Mm-hmm.
And there is this big cell that covers everything.
Oh, I see. Okay. Yeah.
Does,
So, sure.
firstly, sorry,
Go ahead.
does the interface between the stem cell and that big cell that covers everything become messed up in some way? Secondly, do the stem cells fail to coordinate with each other in continuing to generate neurons or
Yeah, this is great. These are great questions. So first of all, the cartoon illustrates that these lineages, a lineage is essentially, I should have explained that better, but a lineage is essentially uh, a clonal unit containing the stem cell and its differentiating siblings. They arrange themselves in these grape-like shapes, which are ultimately surrounded by a cortex glia cell type that keeps everything contained and forms these little units. Now, data I haven't shown you is we have transcriptional data where we compared the neural stem cells specifically across the three conditions. And so one thing that came up is we see a few cell adhesion molecules that are most likely located between the cortex glia and the neural stem cell. So one hypothesis we are thinking about, which we haven't tested yet, is maybe the arrangement of these stem cells, their interaction with these cortex glia, which sort of produce a niche-like environment, could also have an impact on their cell fate and proliferation rates. We have no hard evidence to support this at this point. What was your second question? Whether
2 of 3. In humans and worms, do cells always enter apoptosis if they find themselves to be extremely small immediately after division?
I don't know whether you could say always, but in the studies that produce these data suggest that this is one of the highly likely outcomes.
3 of 3. I go back all the way to the beginning of the presentation. In worms, what is the purpose of doing a cell division where one of the resulting cells immediately dies?
Yeah, that's a hard question to answer. You never know what biology really intended with certain processes. I guess where worms have a fixed lineage, they need to produce a certain number of cell types with a certain final cell fate and function. They apparently implemented a mechanism to make sure that they have the correct number of cells at the end of development. So apoptosis seems to be one of these mechanisms.
Thank you
Yeah,
for
maybe
your
you need an
patience.
odd number for some reason, and you can't generate that with these. Yeah.
Oh,
Yeah.
thank you for your
Maybe. I
patience.
don't know. I have no idea. Yeah. All right. Thank you so much, Kiki. And thank you so much, Clemens, for a wonderful talk and wonderful discussion.
You're welcome.
So yeah, so with that, I would like to thank everybody for coming to the seminar and see everyone in 2 weeks. All right. Take care.